2017, FDA approved the first CAR T therapy, notably Yescarta and Chimera. How did your interest in CAR T therapy really originate?
Well, it goes way back. In the late '80s, I was still studying for a PhD in immunology after having obtained a medical degree. I thought that we should begin to engineer T cells to instruct immune responses. And so my work dates back then. And in the early '90s, we showed that we could introduce a gene into a T cell. And then throughout the next 15 years, our work and that of a few other groups led to the design of not natural receptors, but synthetic receptors that could target T cells to any molecule of our choosing. Those are the molecules that we named chimeric antigen receptors. And the target that we identified, again, many years ago, some 15 years ago, is a molecule called CD19, which is found in most leukemias and lymphomas. And our animal studies suggested that it could be a very good target. Fast forward a few more years, and the first clinical trials started at my center, at Memorial Sloan Kettering Cancer Center in New York, and then at the National Cancer Institute, and at Baylor, and at the University of Pennsylvania. And the shock came then with very spectacular results in a phase 1 trial, which is not the norm. And this has led to the approval of these molecules, as you said, in 2017.
So, what were your thoughts when you really understood that, hey, we have something going here, this is really working?
Well, the first result really came out of animal studies. And you might be shocked to know that those date back really to the early 2000s. We eventually published them in 2003. You might say, well, why? What took so long then? Well, these are not conventional drugs. These are not chemicals. These are not proteins. These are not antibodies. These are engineered T cells. And no one had conducted trials with engineered T cells before. So this is not a discovery in mouse that could then be taken over by the pharmaceutical industry. We had to develop it ourselves, and that means build a facility in which you can handle patient cells, training a team, establishing procedures that had never been established before, and then conducting our own trials. And so that's what led to the approval of these very unique and very unconventional drugs.
So now we know that CAR T has actually revolutionized cancer treatment. So what kind of cancer types are most relevant for this kind of novel approach?
Yes. Well, let me address first the revolution that you alluded to. The approval of these new forms of medicine are first and foremost good news for patients with refractory, relapsed leukemias and lymphomas. For us, though, they also represent the first in a new class of drugs. Again, not an antibody, not a chemical, but a T cell, and a T cell that has been genetically engineered. This is a completely new kind of medicine. The other aspect of this revolution is that, cell therapies have been around, different kinds of cell therapies, mostly in academic centers for many years. But none of them, had previously convinced the pharmaceutical industry to pursue cells as drugs. But this did it. So this is the third aspect, if you like, of this revolution, is that today it is not just academics, but for the very first time, some big pharmaceutical companies that are beginning to develop cells and T cells, and in particular CAR T cells, as drugs. So these are the many effects of this. So the potential, in principle, is large. In principle, you could target any cancer. This cannot be accomplished overnight. One has to identify suitable targets. And as we discussed in today's meetings, there are further improvements that have to be brought to these T cells because some cancers will clearly be even harder to treat than the leukemias and the lymphomas. What's wonderful is to see all the enthusiasm and really this fresh, what I would call fresh blood coming to this field, meaning all the investigators, including in Oslo, by the way, and worldwide, very much in China, by the way, who have now embarked on studying this approach. So we can't promise anything, but what is certain is that there is now a lot of enthusiasm to developing this for other cancers.
So how does this therapy compare to ordinary checkpoint inhibitors?
Yes, so checkpoint inhibitors are sort of the big brother, if you like, of immunotherapy. And what the checkpoint inhibitor does is that in patients who are fortunate enough to have produced their own T cells that can recognize the cancer, the use of checkpoint blockade allows for those T cells that have been blocked, if you like, by the tumor to proceed and do their job. So it's a fantastic therapy. Now, it releases T cells that have been formed in the patient's body, but not all patients have T cells. And so if there's no T cells that recognize their cancer, checkpoint blockade would not work. And we know that as wonderful as it does not work for every cancer and for every patient. And so we think that the role that CAR T cells could play is especially important for those patients who do not respond to checkpoint blockade.
You seem like a modest person. Is your findings, your research comparable to the work of Jim Allison and Bazooka Homeo?
Well, it's for others to decide about, depending on which parameter they use. The impact of CAR T cell at this time is not as large as checkpoint blockade, which has been applied to many cancers. But CAR T cells are also more recent, if I may say, newer. And certainly conceptually, they are completely different. There are many forms of immunotherapy, and the first is checkpoint blockade, and the second one that we see emerging today is engineered T cells.
That's very interesting. I know you have a busy schedule today, and thank you for talking to us, and we look forward to talking to you again sometime. Thanks a lot.
Thank you, my pleasure.